Good morning, everyone. Myself Ishika from BSc Chemistry Honors final year. Today, I will be preparing a topic for visually impaired student from Pradeep's book Bioinorganic Chemistry. Inorganic chemistry deals with primarily with salts and complex metal ions, whereas bioinorganic chemistry deals majorly with inorganic chemistry when applied to biological systems. Thus, bioinorganic chemistry can be regarded as a branch of science that relates inorganic chemistry with life processes by recognizing the role of metal ion in the normal growth and function of biological processes. In brief, bioinorganic chemistry highlights role of metal ions in biology. Bioinorganic chemistry is a rapidly developing area of chemistry because of its importance in biological system. The essential biological roles of a few elements have been known for many years. This chapter will focus on the important role of metals, nonmetals, and their compounds in biological processes. Essential and trace elements. We think that the chemistry of life is solely composed of organic molecules. It is a wrong concept. In fact, there is a long list of inorganic compounds that are essential to living systems. For example, the skeletal and integumental materials outer covering in higher animals is composed majorly of inorganic materials, and the lifetime composition of oxygen by a living entity is a major fraction of its material needs. In addition, water, oxygen, calcium phosphate, and calcium carbonate are the major inorganic constituents of living matter in terms of quantity. Table 5.1 lists the abundance of elements in Earth's crust and in oceans compared with some abundances in typical mammals. Of all the known elements, it is about 30 elements that have been recognized as and essential to life. Of those 30 elements, 15 of them are metals. The essential elements are needed for the growth and normal function of the animals and plants. Absence of these elements results in severe malfunctioning of the living organism. Thus, an element may be considered essential when its insufficient intake produces an impairment of normal function, and its restoration relieves the impaired function. Some of these elements are required in bulk or macroscopic amount to all forms of life and are classified as bulk elements. Some of the bulk elements are required for structural purposes: skin, bones, eggshells, various organs, while the rest are used as minerals. In other words, in the absence of essential elements, the organism can neither grow nor complete its full life cycle. The essential elements can be either subdivided into two groups: bulk elements, essential elements that are formed in higher concentration; trace elements, essential elements that are present in low concentrations. Bulk quantities of group 1 and 2 metals are required mainly to balance the electrical charges associated with the negatively charged macromolecules in the cell, and also to maintain the osmotic pressure inside the cell. The reaction flask of a living organism is the cell. In addition, the transition elements and the heavier members of the group P group also play an important role in the bioinorganic chemistry. Some of the inorganic elements are required in small quantities and are classified as trace or ultra trace element. These different types of essential elements present in the living system are classified in table 5.2. It is important to note that not all the elements displayed in 5.2 are required by all the plants and animals. For example, sodium ion Na positive is of special importance to higher animals. It is, however, not essential to many bacteria and plants except blue-green algae. The trace elements with boron, aluminium, and vanadium are needed by some higher plants. Molybdenum is essential to only those organisms which derive their nitrogen from inorganic sources. Selenium is an essential micronutrient for mammalian species but proves to be toxic at higher levels. Selenium deficiency in humans result in a degenerative condition of heart tissue known as Kishan disease, named after a country in China in which it was endemic because of extremely low selenium levels in the soil, coupled with a population that entirely depended upon locally grown grains. It is important to note that even some essential elements may prove to be toxic if consumed in larger quantities. Essential bulk element sodium and potassium. We have learned that both sodium and potassium are essential for our life processes. For example, we need at least 1 gram of sodium ion per day in our diet. However, because of our habits to adding salt on food, some people consume as many as 5 times more than required. However, intake of potassium is not a problem. Instead, potassium deficiency is common. We must include potassium rich foods such as bananas and coffee in our diet. In organic chemistry, we always think of the similarities between sodium and potassium. However, in biology, it is the difference between sodium and potassium which plays an important role. Cells pump out sodium ions of the cytoplasm, whereas the cell pumps the potassium ion inside. This ion transport is sometimes called a sodium pump, and it involves both the active expulsions of Na positive ion and the intake up of potassium ion. It is the difference in the total alkali metal ion concentration inside and outside the cells that produce an electrical potential across the cell membrane, which is important for the functioning of nerves and muscle cells. The transport of ions require energy, and this is obtained by the hydrolysis of ATP, adenosine triphosphate. It is estimated that hydrolysis of 1 ATP to ADP, adenosine diphosphate, provides enough energy to move 3 sodium ions out of the cell and 2 potassium ions and 1 hydrogen ion back to the cell. Calcium and magnesium. Both calcium and magnesium are present in body fluids. Magnesium ions are concentrated in the animal cell and calcium ion are concentrated outside the cell. This is similar to the presence of potassium ion inside the cell, whereas sodium ion outside the cell. Calcium ions are present in the external skeletal of creatures such as shellfish and corals. The material used is calcium carbonate. Creatures with interior skeletons such as mammals and reptiles use calcium hydroxide phosphate Ca5OH PO4 whole thrice, and the animal on teeth of fluoroepitete 3 into Ca3PO4 whole twice CaF2. Both these compounds are highly insoluble. Important minerals used in biological materials are listed in table 5.4. Calcium ions are important in blood clotting and are required to trigger the concentration of muscles, such as those that control the beating of heart. In fact, some types of muscle cramps can be prevented by increasing the intake of calcium ion. Calcium containing minerals in biological materials. Calcite CaCO3, argonite CaCO3, and bird's eggshell, molluscus shell, vertebrate bones. A decrease in Ca2 positive content of blood, either due to dietary deficiency or from an insufficiency of the parathyroid hormone, may lead to disease called tetany. On the other hand, an increase in the concentration of calcium ions in the blood may lead to calcification of several internal organisms. In absorption of dietary calcium ions from the intestinal tract may be prevented by the simultaneous ingestion of organic acids, example oxalic acid, with which it combines to form an insoluble salt. It means that an availability of dietary calcium is greatly affected by the other constituent of the diet. Both calcium and magnesium ions are excreted mainly via the large intestine instead of the kidney. Magnesium, because of its high charge density ratio and consequent strong hydration, as in MgH2O whole 6 2 positive, plays important biological roles that are different from those of calcium. It acts as a counter ion to the negatively charged group in nucleotides and polynucleotides. One of the most important nucleotide is ATP, adenosine triphosphate, which serves as the fundamental source of the living cells. ATP is thus known as energy-rich molecule and is used to derive most of the chemical reaction of the cell. Mgt positive ions form a complex with ATP. It has been recorded phosphate. Phosphorus is an important element of for life processes. For example, free hydrogen phosphate PO3 negative and dihydrogen phosphate involved ions are involved in the blood buffering system. Phosphate is the linking unit in the sugar esters of DNA, deoxyribonucleic acid, and RNA ribonucleic acid. The phosphate units make up a part of ATP, the essential energy storage unit in living organisms. Bone is a phosphate mineral. The commonly called mineral's epitide is calcium hydroxide phosphate. The presence of phosphorus in bones and its function in regulation of pH of blood constitute important features of phosphate. It is important to mention that the normal formation of bones depends upon the relative amount of dietary phosphorus and calcium in addition to vitamin D. Essential trace elements. Among the known trace elements, the three most prominent biologically active metals are iron, copper, and zinc. The remaining trace elements, excepting boron and fluorine, are used in very small amounts, less than 10 mg in adult humans, and are referred as ultra trace elements. The trace elements are known to play important role as metalloenzymes. Iron is essential in small amount for both plants and animal life. However, like copper and selenium, it is toxic when used in large quantities. Biologically, iron is the most important transition element and is found in greatest abundance in biological system in the form of porphyrin complexes or hemes such as hemoglobin or myoglobin. Porphyrins are macrocyclic ligands and are derivatives of porphyrin, discussed later. A human body weighing about 70 kg contains 4.3 gram of iron. Nearly 70% of this iron is present in hemoglobin and about 3% of myoglobin. The remaining 23% of iron of body iron is present as ferritin. Ferritin is present in spleen, liver, and bone marrow. It consists of a shell of linked amino acid peptides called apoferritin surrounding a core of an iron oxohydroxophosphate. The core is a cluster of iron 3 ions, oxide ion, hydroxide ion, and phosphate ions with the composition FeOOH whole 8, FeOPO3H2. It is very large, containing up to 4500 iron ions. With its hydrophilic coating, this large aggregate is water soluble and is concentrated in the spleen, liver, and bone marrow. Apoferritin protein can be prepared from ferritin by reduction of ferric ions to ferrous ions at pH 4.5, followed by the removal of iron by dialysis. The formula weight of apoferritin is nearly 46,500. Ferritin can also be synthesized in laboratory with properties similar to natural ferritin. It can be prepared by treating crystalline apoferritin with an inorganic iron salt in the presence of oxygen. Ferritin is probably formed in vivo by an analogous process. The various steps involved in the pathway of iron from blood stuff to hemoglobin and to ferritin involves the following process. The Fe2 positive of the bound dietary materials is reduced to Fe2 positive in the gastrointestinal tract. After absorption into the cells of the intestinal mucosa, Fe2 positive is incorporated into ferritin as Fe3 positive. The Fe2 positive in mucosa is also converted to Fe3 positive plasma bounded by iron binding globulin named transferrin. Plasma 3 positive is in equilibrium with iron in the liver, spleen, and bone marrow. Zinc. Among the trace essential elements, zinc is the second only to iron in importance. Zinc is essential to all organisms and plays an important biological role in the enzyme system of animals and plants. Humans contain about 2 grams of zinc and is one of the most important and crucial element in our diet. Deficiency of zinc are not life threatening but contribute to fatigue, lethargy and related symptoms. Zinc ion is a soft acceptor compared to other divalent ions and act as a stronger Lewis acid towards many biomolecules. Compared to zinc ions, copper ion is also soft but has an additional intermediary oxidation state that is copper positive. Different oxidation states of copper may allow different redox reactions and complicate its behavior. The complex of zinc ion are much more labile than the corresponding complexes of magnesium, nickel, and magnesium. This facilitates the role of zinc ion in enzymes since zinc ion is completely resistant to redox changes at biological potential. Its role cannot be affected by changing the redox potential in the organisms. A number of enzymes containing zinc ion are known. The most important of all the enzyme is the carbonic anhydrase. It is present in red blood cells and is involved in the respiration. It speeds up the absorption of carbon dioxide by red blood cells in muscles and other tissues. On the other hand, in the reverse reaction, it involves the release of CO2 in the lungs. The reaction is pH dependent. CO2 plus H2O in presence of carbonic anhydrase gives H2CO3. Thus, the zinc containing enzyme carbonic anhydrase catalyzes the otherwise slow carbonic acid carbon dioxide equilibrium reaction. The carbonic anhydrase has a common 4 coordinated environment around Zn2 positive ion in which 3 of the ligand are imidazole nitrogens of 3 histidine and the fourth is a water molecule or hydroxide ion. The water molecule coordinated to a zinc ion has a pK near 7 and thus the role of zinc is to reduce the basicity of hydroxide ion such that a high effective concentration of hydroxide can exist in an approximately neutral medium. Another primarily enzyme containing Zn2 positive ion is carboxypeptidase. It is important in the pancreatic juice and is primarily involved in the digestion of proteins by animals and protein metabolism in plants and animals. This enzyme catalyzes the hydrolysis of the terminal peptide link at the carbonyl end of the peptide chain. The enzyme is selective and works only when the R group is the terminal amino acid and is aromatic or a branched aliphatic chain and has a L configuration.